Welcome, everybody. Thank you all for coming to this webinar co-hosted with Cederlane, our partner in Canada. My name is Irene, and I work as the Cytognos Sales and Support Manager for Canada. And I'm presenting today your cheat sheet for smarter clinical flow cytometry testing. So I have Jessica from Cederlane with us today. Hi, Jessica. How are you? Thank you for joining.
Hi, thank you, Irene. I'm gonna. Oh, can you hear me? Okay.
Yeah.
Okay,
no problem.
Awesome. So thank you. Hello, everyone. Thanks for joining today. My name is Jessica. I'm the diagnostic product specialist at Cederlane. I manage our clinical supplier portfolios and work with our clinical customers in various labs, including flow cytometry, pathology, microbiology, infectious disease, and so on. If you need quotes, tech support, help sourcing products, please contact me. I'll be happy to help you. You can reach me at the details on this slide. For anyone not familiar with Cederlane, we're a private company in Burlington, Ontario. We've been in business for more than 60 years. We manufacture immunology products, including antibodies for flow cytometry, cell separation media, FLAIR for PNH testing, and so on. And then in addition to manufacturing, we also distribute for many companies, which are listed on your website for your reference. We've built many long-term relationships with our supplier partners, and we're proud to be partnered with Cytognos in Canada. So I hope that everyone enjoys the webinar today. Thank you.
Thank you, Jessica, for that introduction. Okay, so now we're ready to start. These are the points that we are going to cover today. First, I will start off highlighting the value of flow cytometry in clinical practice. Then I will talk about the current issues we face in the clinical routine lab, and I will focus on the specific challenges of the minimal residual disease assays and on the design and validation of flow cytometry tests. Finally, we will see how we can overcome these challenges with a fully standardized solution, the EuroFlow method. And at the end of the webinar, we'll have a few minutes for questions. So, what is the role of flow cytometry immunophenotyping in the clinical lab? It allows the diagnosis and classification of leukemia, lymphoma, and plasma cell dyscrasias, and the detection of minimal residual disease after treatment. Flow cytometry is also used for studying the immune response of patients and for progress control of the treatment itself. And in the clinical immunology field, to support primary immunodeficiencies diagnosis. Now, flow cytometry is definitely a versatile and powerful technique with many applications in clinical settings, but it's also complex and it has multiple steps. First, we collect the specimen, transport it to the lab. Once it gets there, we process the sample, we stain it, and acquire the tubes in the instrument, which has been previously calibrated and compensated. And last step is analyzing the data and reporting the results to the clinicians. So there are multiple checkpoints here where we can introduce errors, especially in the pre-analytical and post-analytical phase. And these errors have economical consequences and negative effects in patient care. And that's why all flow cytometry assays require careful assay design, instrument setup, performance validation, and ongoing quality monitoring. Now let's see what are the common challenges we face in clinical routine. First, sample stability can be an issue. Most of times we require a fresh sample, in some cases no longer than 36 hours after collection. The samples that come in are very diverse. We get blood, bone marrow, lymph nodes, and they come with different anticoagulants like EDTA, heparin. So they require different shipping and storage conditions especially with regards to the temperature. There are different work-life protocols in place, so we have to coordinate multiple workflows at the same time. Cocktailing the single antibodies can lead to pipetting errors, and if we get a new lot, we need to cross-check it with the previous one. And we need to have an efficient stock control of all the reagents to reduce reagent waste. Now, our instruments have to be correctly set up, and we need to track the day-to-day performance to ensure consistent data over time. It's important to acquire the tubes as soon as possible to minimize sedimentation and not to compromise the quality of the sample. And during the acquisition in the instrument, we can run into issues such as unstable flow, clogging, or air bubbles. When it comes to the analysis, there is always a user bias because positive and negative populations are clear, but there is a lot of middle ground in between. So the final results highly depend on the analyst expertise. And lastly, we have to put together a report with our lab test results, transfer it to the LIS system so it can be accessible to the pathologist and the clinicians, while ensuring that all the sensitive data is protected. So these are the common points regardless of the type of study. However, more sophisticated tests, such as minimal residual disease assays, pose additional challenges in the lab. To follow up patients, we usually analyze bone marrow, which is a very delicate specimen. Sometimes we only get a small volume to run the test, and we can have hemodilution issues if we don't get the first pool. Plus, the anticoagulant may affect marker expression. For example, heparin downregulates the CD138 expression in plasma cells. Then we have to bulkize the sample upfront to concentrate the number of cells. So we need to add an extra step to our sample processing protocol. We need to make sure that our MRD panels are optimized for detecting the target population at very low levels, and that the panels are adapted to immunotherapies that can interfere with the expression of certain markers. And definitely, we need to make sure that our instrument has the capacity to collect a high number of events relatively fast. One of the biggest challenges in MRD assays, as we know, are the false MRD negatives, because we are handling a large number of events, and we want to identify a population that has a very low frequency. So high sensitivity is key to avoid reporting false MRD negatives here. Plus, we need to consider the patient treatment for the gating strategy. Some immunotherapies can mask the expression of certain markers. On top of that, in every pathology lab, we are getting more and more cases. We have a very tight turnaround time. Because of the COVID-19 pandemic, hospitals may have cut the budget for the lab. And at the same time, we need to meet all the regulatory requirements for diagnostic labs. So we can summarize all this in one sentence. In a clinical lab, we have to get results out quickly without compromising quality. Now, before introducing a new test in the lab, we need to carefully design and clinically validate the assay. And to design a new flow test, there are several aspects we need to keep in mind. First of all, the clinical question that we want to answer. And the clinical question is going to determine the panel design, the sample processing protocols, the instrument settings, the data acquisition parameters, and the data analysis strategy. Once we come up with the antibody panel, we need to test the entire assay. And this is an iterative process that can take many, many rounds of design, redesigned before we complete it. After we are done with this testing phase, we need to ensure that our assay complies with the current in vitro diagnostic regulation. And we need to put together a fit-for-purpose validation plan and assess several parameters like specificity, precision, sensitivity, which is essential for MRD assays, carryover, and even establishing reference ranges. Just to let you know that to date, there are no specific guidelines for validating flow cytometry tests in regulated environments, which makes the entire process more complicated. The question is there a way we can get consistent results over time across instruments and operators, save money and time while we comply with regulatory bodies? The answer to this question is yes, by standardizing all the steps in our clinical routine. And that's exactly what the EuroFlow method is about. The EuroFlow Group is an independent scientific foundation made up of over 20 companies and research groups, which was born in 2006 with the mission of developing, standardizing, and validating highly sensitive flow cytometry tests for diagnosis and follow-up. The EuroFlow method is a screening approach looking to use fewer antibodies at first to narrow down the malignancy, and those are the screening tubes, and then running classification panels for further differentiation. They started with leukemia lymphoma panels, followed by MRD panels to monitor patients. And once they got the system in place for onco-hematology, they applied all their expertise and knowledge for the primary immunodeficiency and the immune monitoring fields. This EuroFlow method can be used in the onco-hematology, clinical trials, and clinical immunology areas, but today we are just going to focus on the onco-hematology. And this method includes the SOPs, the antibody panels, the analysis software tools, and a QA program. So now let's go over each of them. There is a protocol for bone marrow sample collection and transport, specifically for myeloma MRD testing. And also there are guidelines on sample collection for regular testing. What else? There is a protocol for sample processing and staining, including extracellular and cytoplasmic antibody staining guidelines. There is a specific bulk lysis protocol for MRD testing. For building leukemia, lymphoma, and myeloma panels, the EuroFlow tested a large number of fluorochromes and antibody clones, and they selected the fluorochromes based on the MFI, the stain index, the spillover, the stability, and the availability. Here we have an example illustrating the comparison between two fluorochromes, the APC H7 and the APC C750, conjugated with the same antibody clone, the CD10, and tested in the same sample under the same conditions. What we see here is the APC is that the APC C750 conjugate shows a brighter MFI, which provides a better resolution between the positive and the negative reference populations. Likewise, for selecting the markers, they looked at the antibody clones, the conjugates, the number of combined markers in a single tube, and the amount of antibody and volume of staining. They tested the performance of the marker combination together using software tools, and that's what we can see in these diagrams. So these are multidimensional plots showing cases of T-ALL in green, BCP-ALL in blue, and AML in red. And all these cases were stained with the EuroFlow acute leukemia screening panel. Now, in each plot, they left out one of the markers of the panel in order to study one by one its discrimination power of these three acute leukemia subtypes. And these results indicate that each individual marker included in this panel is essential because missing one of them affects the identification of the different acute leukemia entities. Now, there are protocols for instrument setup, calibration, and compensation available for different instruments, for FACSCAN2, Navios, and FACSLyric as well. And these protocols cover the instrument configuration, the setup of forward and side scatter, the setup of PMT voltages for target fluorescence channels using rainbow beads, the compensation settings, and the instrument performance monitoring. For the data analysis, we need flexible and advanced tools that make the multicolored analysis easier. For example, by turning off the background so we can just focus on the target populations, by looking at our data in many different ways, and by being able of studying the cell maturation path of a specific population. Also, we need tools to simplify the multi-parameter analysis. As flow cytometry technology evolves, there are more and more parameters to analyze. In this slide, we have all the possible combination of markers in an 8-color panel, and that's a total of 64 diagrams. And instead of looking at all these 2D plots, we can convert all of them into one multidimensional diagram that separates out populations using all the parameters in our panel. And this way, we can discriminate populations with a very similar phenotype that can go unnoticed if we just look at the 2D plots. We can check the complete immunophenotype of a target population versus a normal control. And we can follow up patients with confidence by loading the diagnostic population onto the MRD case as a reference. We can analyze patient cases and not single tubes, which is very important, because we can combine all tubes from the panel, analyze them together, and create one single report. Okay, still the problem in flow cytometry analysis is the lack of a good reference point or baseline. And as complexity of flow panels and data analysis increases, we really need big data analysis approaches. So we need automation for our clinical flow analysis. To put it simply, the automation consists of clustering all the events in the file, followed by an identification of all these clusters. But to be able to give the populations and clinical and biological meaning, we need to feed these algorithms with human knowledge and expertise, and that's the EuroFlow reference database. Okay, let's see an example of automation in acute leukemia screening. So we get all the cell subsets in our sample identified and characterized. And also we get a compass diagram pointing out the cell lineage, the T, B, or myeloid lineage, that the aberrant cells are more likely to belong to. This is basically done through a multiparametric comparison of your sample versus a reference database. And that reference, the reference database in that particular case, contains B leukemic cells in green, T leukemic cells in blue, and myeloid leukemic cells in red. Here we have the interpretation of the case, including not only the blast information, but also the detection of potential related disorders of the patient. And we get the complete immunophenotype of each of the aberrant populations in case that we have several lineage affected. And additional information on the present or missing populations of the sample. And what is more important, we also get information on which panel to run next for further classification of the disease. Okay, that's for the acute leukemia screening. Now let's see another example of automation in myeloma MRD. So here we can see the identification and characterization of both normal plasma cells and abnormal plasma cells after automation. And here we have the interpretation of the case, including a complete immune profiling of the patient versus a normal reference, information on the bone marrow regeneration, the MRD level and the immunophenotype of the aberrant population, the clinical threshold that determines the sensitivity of the test, and information on sample quality issues like hemodilution and sample viability. Okay, going back to the steps of a flow cytometry assays, EuroFlow runs 2 external quality assessment programs to certify the consistency and reliability of results generated in the lab. So we have the LST QA that monitors the overall flow testing performance, including the sample preparation, the instrument settings, and the data acquisition. And the MMMRD QA that specifically targets the data analysis of the myeloma MRD assay. The EuroFlow goal is to provide a standardized and validated, highly sensitive flow cytometry test for diagnosis and follow-up. And that requires a close collaboration between the industry and the academia. So thanks to this collaboration, Cytognos can bring the EuroFlow method as a ready-to-use flow solution for any lab in the world. And this is what we call next-generation flow solutions, as they include the laboratory SOPs, the dry down antibody kits, the OmniSite instrument, the InfiniSite software package, and ongoing training and support to help you along the way. To wrap up, these are the key takeaways from the webinar. Flow cytometry is a very powerful and versatile tool in clinical practice, and yet complex. Standardization is the way to obtain reliable and reproducible results. And the EuroFlow method is available through Cytognos as a ready-to-use solution for your lab. Just to let you know that if you have any question or you want more information about our next generation flow solutions, feel free to reach out to us anytime. And that's all. Thank you so much for watching. And now it's time for questions. So I will be happy to take any question that you have.